Hey, this is MCB2 tutoring again. So this is the second part of the lipoprotein videos I'm going to do. So last one we talked about chylomicrons, and then this one we're going to talk about the VLDLs. So recall the chylomicron job is basically delivering the triacylglycerides to the periphery, and it came from the GI. So it's delivering it from the GI to the periphery, and then what's remaining is going to end up in the liver. So remember, the liver is able to do many things, and one of them is it does lots of fatty acid synthesis. So if it does the fatty acid synthesis, eventually, fatty acids, it's going to combine with some glycerol, and that's going to result in the formation of triglycerides. And since you're making the triglycerides, it's going to need to deliver that to the periphery. And how it's going to do that is in the form of these lipoproteins. So before I go into the VLDLs and other lipoproteins, I just want to mention one more thing. So remember, we're talking about, well, the lipids are kind of hydrophobic, right? So they need to be delivered in the form of lipoproteins. But how hydrophobic these lipids are also depends on the size of their chain. So the shorter to medium chains, can be delivered by albumin. So over here, I'll just write, so basically, okay, so the short or medium chain fatty acid is delivered by albumin. And then the long chains fatty acids need the lipoprotein. So just keep that in mind. This same thing sort of applies to the digestion as well. So the shorter and medium chains, they just diffuse in. They need to be cleaved off of the triglycerides, but then they'll just diffuse right into the cell, and then instead of going to the chylomicrons, they're also transported by albumin. So just keep that in mind. Okay, so now in the liver, we're talking about like fatty acid synthesis, and then it's made into these triglycerides. But these triglycerides are, pretty big and hydrophobic, so it requires lipoprotein to deliver them. And the lipoprotein here is also going to have an apoprotein. So recall, it is also made from this ApoB gene. So B, apoprotein gene. It has two possible products. One of them is an ApoB48. That's only in the GI for chylomicrons. But in the liver, it's going to be made into ApoB100, and that's for the VLDLs. That's one. Okay. And to assemble these together, same thing happens as the GI. So these are going to be made in the rough ER, and the triglycerides are basically going to be put together in the smooth ER. So this needs to be combined into the ApoB100. So this transport of triglycerides into this ApoB and then kind of combined into this lipoprotein, this is mediated by MTP. MTP stands for microsomal transfer protein. So it's the same process. It's also mediated by MTP. Okay, so this is going to exit, and this becomes the VLDL. So So that's very low density lipoprotein. Okay, so I drew another one here, so it's the same guy, VLDL. Just like the chylomicron, the main component of this is the triglycerides. And then here it's made of like somewhere around 50% triglycerides. And, you know, just like the other one, it has some cholesterol, and it has some cholesterol ester. And it's going to do the same thing, very similar to the chylomicron. The only difference is that it came from the liver. But it's going to do the same thing. It's going to, you know, drop off some triglycerides. And when that happens, it's going to become slightly higher density. So that's an intermediate density lipoprotein. And then this one drops off more triglycerides and then becomes a low density lipoprotein. And just like over here, where the HDL had to initially give it a few apoproteins, so HDL here will also be giving this VLDL some apoproteins. So notably, this is ApoC2 and ApoE. This is exactly the same step as the chylomicrons, so the similarity here. And, you know, on the surface here somewhere is a ApoB100 that I got from the liver. Okay. So the process of dropping off the triglyceride also requires the lipoprotein lipases. So in order for this to occur, you also need some lipoprotein lipases that are located on the blood vessel wall here in pink. Just like the other one, since it got the C2 from the HDL, these VLDLs and IDLs, they all have this ApoC2. And what it does is it activates the lipoprotein lipases. Okay, so this is fairly similar, very straightforward. It's very similar to the chylomicrons. But the main difference here is in the VLDL. So VLDL differs in the sense that instead of going into the liver, it's going to be taken up by the peripheral tissue. So let's talk about, okay, so the triglycerides has dropped off so much triglycerides, so now triglycerides is only a very small portion of this LDL. Sorry, it's like a tongue twister. And it's going to have some cholesterol, and it's going to have some cholesterol ester. And recall, it's still going to have the ApoB100 that was made by the liver. And the peripheral tissue needs to have some sort of receptor for the LDLs, right? It needs to have some kind of LDL receptor. And that's perfect because ApoB100 is right there on the surface, and then this LDL receptor actually recognizes the ApoB100 that is supposed to be on every single LDL. So the ApoB100 is going to bind to the LDL receptor, and then the whole thing is going to be endocytosed. Okay, so now you will have a lot of cholesterol inside the cell. And the cholesterol, basically the cells don't want to have the cholesterol maintained in cholesterol form, and what they want to do is that they actually want to change it into cholesterol esters. The reason being, cholesterol is pretty much a component of every single membrane. So the membrane of these peripheral tissues have cholesterol, right? And any of the membrane-bound organelles have cholesterol. So if you just keep the cholesterol around, that's going to interfere with the fluidity of all those membranes, and that's going to interfere with, basic cellular function. So cholesterol, you have to change basically the cholesterol into something else that will not be inserted into the membrane. And the only reason cholesterol is inserted into the membrane is that, well, let's just pretend these are the 4 rings of cholesterol. They're not organized like this. This is just the very, very general diagram. Okay, so cholesterols have this OH group on it. Okay, it has like 4 rings, something like this. And then okay, sorry, this is not working. Okay, so the cholesterol ester has this basic shape. It has 4 rings, but that's not what we're talking about right now. We're concerned with this little group here. So the cholesterol esters has this group. And basically what happens is that this whole molecule is it's entirely hydrophobic. As opposed to cholesterol, it has a hydrophilic side, and then hydrophilic, and then this part is hydrophobic. So cholesterol, because it has it has one polar and one nonpolar side, this is able to insert into the membrane, whereas cholesterol esters, being all hydrophobic, will be kind of contained in its own little droplets. So that's not going to interfere with membrane function. So it's important that all the cholesterol in the cell is stored in the form of cholesterol ester. So to catalyze this very important reaction is this enzyme called ACAT. And it's needed to be able to convert cholesterol to cholesterol ester. Okay, so that's one of the things that happen once cholesterol is internalized. The other thing is cholesterol mediates 2 other reactions. Well, not 2 other reactions. It regulates 2 other events in the cell. So basically, if you have a lot of cholesterol, it's an indication that you probably don't need to make more cholesterol. So you would imagine in cholesterol synthesis, it's going to inhibit cholesterol synthesis. So remember, the enzyme that's really important for cholesterol synthesis, the highly regulated step, is the HMG-CoA reductase. That's involved in the cholesterol synthesis. So if you have a lot of cholesterol, you don't want to make more cholesterol. So cholesterol will inhibit the action of HMG-CoA reductase. And the same idea, if you have a lot of cholesterol, you probably don't need to uptake more cholesterol from the extracellular space. So this LDL receptor will also be downregulated by the presence of lots of cholesterol in the cell. So these 3 things will happen as a result of increased cholesterol. Okay, so this is basically the story with VLDL. So VLDL came from the liver. It has an ApoB100, but then it will acquire an ApoC2 and ApoE from an HDL. And the goal of the VLDL is to basically drop off triglycerides to the peripheral tissue. So it'll continuously do that until it has so little triglyceride and it becomes an LDL. An LDL has a copy of ApoB100 that will bind to the LDL receptor. And the LDL receptor will take up this LDL, and then the cholesterol will go into the cell. So this is the general pathway for the VLDLs. The next video, we'll talk about how HDLs play into this.